Thank you so much. I know I'm the last person to talk, so I should try to fall or something so people laugh a little bit and wake up. But it's just humbling to see what has happened in the last year in the field of COL4A1. And I was asked to talk about exactly the question that was posed to Dr. Granata, like what do we do to get therapies to the clinic to improve the life of these patients? These are my disclosures. I'm funded by NIH, several agreements. I do consult for a lot of biotech and pharma because we're trying to bring these therapies to clinical trials. But it's not without the foundations that we start the work. There has not been a disease that we have tackled, even to get to FDA approval, that hasn't started with the foundation support. So to emphasize the work of this foundation and the foundations that are being formed at the moment. This is where we are physically. We are in Boston, Massachusetts, in the United States, East Coast. The hospital hosts inside the general hospital, very large, a small pediatric hospital. So we are a small pediatric hospital inside a large hospital, which allows us to provide care and also do research and investigation from before birth all the way to our end-of-life care, whenever that is. So I'm going to try to walk you through, and I'm going to use this little schematic throughout the talk to come back and come back and come back. But this is what we call it, a genetic research cycle. I don't care what it's called. To me, there is no therapies. It doesn't have to be linear, but we do need to understand the most critical aspects of this. And it starts with the families and the patients. This was a question asked, but what does it take to get a drug approved by the FDA? So it was very well said by Dr. Granata. If it's an approved drug that you use for another disease, that's called repurposing. And still, nobody will be able to get it paid by insurance, which doesn't matter in certain countries where there is public health care, maybe. But it does matter for a lot of them if you don't do a clinical trial. So the clinical trial usually is still a requirement to have an added indication to a drug that is FDA approved. But let me tell you something. In pediatrics, we have 7 FDA-approved drugs. The rest of all the medicines that your kids take every day are not FDA-approved for pediatrics. They have been used by clinicians with rationale that they are safe, and then collective data and many publications like the ones that we are seeing being done here by all these collaborators and clinicians is what makes the drug indisputably have to be used or can be covered by insurance. So I'll use this schematic for what I'm relating here as targeted gene therapies, so trying to go to the source problem, which is the genetic mutation, and I'll tell you the different places. But all those presentations that we heard about today, from animal models of disease, meaning zebrafish or mouse, or the cell assays that we were looking at, like iPS cells or fibroblasts from patients, or cells outside the body of a mouse, like Scott was relating to us, all of that is called preclinical development. So until we touch a human, that is preclinical. When we touch a human, we call it a clinical trial, which means that's when we are putting an intervention inside the patient. But this thing here that you see kind of happens usually, hopefully, in parallel with this, because as you heard, this takes several years to get done, is what we also call clinical trials. This is patients participating in different types of research. And you've heard, like, since the Italian first conference before last year to this year, and what has been put in progress by different groups with a lot of patients is cross-sectional studies. So you grab what you have at that point. That's one point in time, and you just try to see everything you can understand and correlate with disease. And usually this is enough to start interacting with the regulatory agencies, both the European and the FDA, and every country has their agency, by the way. The European Union has one. The US has another one, which covers North America. But if you are developing the drug for India or for Latin America, you have to deal with the regulatory entities of those countries. And why do we interact that early? And in the FDA, this is called Interact. Meeting is to educate them. And we do have to do that for COL4A1. There has been no Interact meeting for any therapeutic of any kind for the FDA. In fact, the FDA has a mechanism called the voice of the patient, which is how you just collect, foundations usually are who move this and inform the FDA on the burden of the disease. That's a very professional interaction. It's a whole day interaction with the FDA, gets recorded and made available to the world. And you can collect witnesses of how the disease affects your life. Children, adults, parents, caregivers, educators. We've done it for several diseases. That allows us to have the entities ready to speak about the disease when we bring the therapeutics. Remember, these are complex diseases, and they're also rare, so they don't need to know about COL4A1 for a lot of reasons. Then we do these retrospectives. This is not very different, to be honest, than the cross-sectional, but retrospective is usually more in-depth studies. You heard today about several retrospective studies done on the clinical databases of countries like France, Italy, and even in centers. So the patients that are coming, like Rachel was talking about at UCSF with their cohort, our cohort, Diana was talking about. Elizabeth's group has presented larger comparisons and much more in-depth presentations. And we started the day with this. We started the day with, like, what are the presentations of patients with these mutations? Seizures, no seizures, cortical malformations, do they bleed, do they not bleed? This is critical. And these ones, by the way, and this is maybe for tomorrow, we can do a lot of this by patient directly providing medical records anywhere in the world. So if you consent to a protocol in one institution and the patient in the consent agrees with what are the conditions of sharing and all the things and recontacted or not, et cetera, there are different parts of the consent. You can provide your own medical records and help more a center or anyone that is centralized to get all these records. If any effort is being done for registry or real databases, we should all be trying to advocate that all our protocols, I don't care where we are, have a direct patient consent that allows us to be shared, and we can collect the medical records. What it takes resources is to get the stuff out of the medical records. Medical records don't necessarily speak to databases like the one that we heard today, and it takes a lot of effort curating the data inside those medical records. Months and months of work by research coordinators, clinicians, radiologists are necessary to get the data out. But can a patient from anywhere in the world be included in our protocols? Anywhere in the world, the answer is yes. And this is legally a frame of research, which is not the same for clinical. Prospective studies, now it's complicated and very expensive. When you have to go to the same place to get your MRI every year, and then you're doing several evaluations, you need to travel there, and you need to get all of this covered. And as Rachel was saying, she opens the natural history studies. Patients are eager to come, but there is a lot of restrictions and limitations if there is no coverage by healthcare system or insurance systems. In Europe, the boundaries are there. They're a little bit more foggy, but I think they're still country-wise quite limiting. So we do have to get robust funding to do a prospective natural history study. You want numbers? 1.5 to 2.5 million for a 3-year study for 14 patients in the US. That's probably half in Europe, okay? And what does it give you? It gives you the most stringent database that you're gonna have in the disease. Would it work for COL4A1? Only if we grab one cohort. If we grab everybody, all comers, we're gonna have a dilutional effect of what kind of things. The disease is different in babies than in teenagers, and it's different even with the same mutation in a single family. You guys know this. So you have the same mutation. There are relatives of yours who have not had any issues or have not had symptoms that are clinically identifiable. I think Scott brought in the fact that we are not looking in cognitive decline very deeply in COL4A1, so there might be more symptoms that we know of. Another reason why to do the prospectives, that's when you do the batteries of these psychological and neuropsychiatric evaluations, which also allows you to develop clinical guidelines to do this. And all of this, hopefully, as I was saying, is happening at the same time. It's absolutely necessary to get to this thing called here IND for the FDA. It's called the same pretty much in the EMA, which is when I have a drug, either if I want to repurpose it, you don't need an IND if you're repurposing, but if you have a new drug and you want to get an approval to try it in humans, to do the first in-human studies, they have to approve you. And why do they approve you? To make sure that what you're doing is rational, safe, and justifies the risk-benefit. The idea is not letting anyone do garage science and getting kids, babies, to get a therapy that could harm them in a very irreversible manner. So we can bitch about this and how much work it is and how intense it is, but it's really for the protection of human health that you're having these regulatory agencies be there. And that's not where it stops. It is still an experiment. We heard today about experiments everywhere, no? In the cells, in the zebrafish, in the mouse. These are experiments in humans, and they're experiments. We don't know if they work until we get the data from the clinical trials. So patients put a lot of things at risk when they participate in this because we don't know the risk until we do it for the first time in humans. If the efficacy is there and the safety is acceptable, things are reversible, you get nausea, vomiting, but you get better, and then you got a much, much more robust effect from the drug. The regulatory agency, the A here is from FD, the flags that were regulatory took out FD, and then you will be able to get a clinical trial and an approval. So when I say FDA approved drug, now there is a mandate by the healthcare system to provide it. And I'll give you an example when that mandate falls through, and this happened just to the first gene therapy approved for leukodystrophy, which was approved in Europe by the EMA, but the council that had to regulate how much it cost and whether countries are gonna provide it decided not worth the cost for the benefits and for the population. So it's not commercialized in Europe. No patient in Europe can access it in Europe because it's not sold in Europe. So you had to move to a country where the drug is approved. So it doesn't end with approval. So this is what I'm trying to say is a lot of things need to be done at the same time to make sure that the drugs are affordable. If we are developing drugs that cost $3 million each, we're gonna have a problem. And it's gonna only be for a very selective number of people, which is not where I come from and not where our mission it should be. It should be treatments for everybody. So let's go into, you start with the community. We already started, okay? This is a much more organized and beautifully organized community. And thank you to these two women in that chair over there. It started in Italy way before any of us, except Doug, of course, that has been involved, I think, from the creation of everything. But it started with a European effort, with a local effort, a local family, a local country, a local group of physicians getting together. And what happened before the first conference happened last year is already incredible. And we were discussing today, this conference is dramatically different than the one last year. Last year, it was almost like a recap of the Italian consensus. This conference has expanded, and the most beautiful thing to see, the engagement that you got in 12 months in your disease that is rare. So I cannot emphasize enough how much this needs to be supported by everybody. All stakeholders come and work together on advocacy groups. I give a shout to the renaissance of the Gold Syndrome Foundation. Team, feel the weight here, because you're gonna have to bring back the messages, but there is a lot of work to be done, and I know there is a French group. We were looking for website and pictures and stuff, but we weren't able to get it, but we'll be, exactly, so this is happening, and it's happening soon. What I like about all of this, that these new organizations are already working together. Let's not duplicate efforts. Pick parts of the mission that everybody can do is in their warehouse, in your capacity, in the heartbeat of your patients and your families, and take on one of those missions. There is so much to be done. So description of the phenotypes. You heard this word phenotype. If you don't know what that is like what kind of symptoms that the disease gives you. That's what a phenotype, okay? So you heard today there are some kids that have seizures, others don't. There are adults that have cognitive problems, the others don't. Some people get strokes, different kinds. That's phenotypes. We got complicated in COL4A1 because it's not just the brain. Collagen is everywhere. Thank goodness other cells of the body do compensate, as Dr. Gold was telling us. So we do have a multi-system disease, and this is where the patient voice comes into place. We're not gonna be able to treat and say, oh, I corrected everything, and everything is much better, because I don't even know, Rachel was telling us today, we don't know about GI symptoms, and we don't even know where they're coming from. If it's the disease, if it's the other parts, if it's the CNS, if it's the white matter injury, we don't know. So let's start with the things that we do know, how much the cerebrovascular function and dysfunction alters the quality of life of these patients. And we do know that part. We know something about the hematological system, we know about the musculoskeletal system, I could not summarize Elizabeth's paper, so I put the first authors of everybody in the last few years, just to say that there's been a lot of work in getting the voice out there of what the disease is doing and what we call genotype-phenotype correlation. But it's very complex. You just heard today how same mutations can give you different diseases. So this is one of those phenotypes. We always refer to GOLD syndrome. It's mostly referring to this early congenital phenotype, which can have these severe prenatal hemorrhages, most commonly the porencephalic cyst. You heard all of this about. I'm not going to repeat the talks that were incredible, and they're also going to be recorded. So if you want to go refresh your memory, you can do this. And these are courtesy of Doug. He helped us. We were putting some words together and trying to get some more funding, and he helped us put a couple of these slides together. But this is more than the congenital disease, and you heard about, for example, patients with white matter disease presenting in adulthood. This is a patient that had a small stroke, like hemiparesis, difficulty speaking. But years go on, even with this white matter injury, and this is in Boston. This patient is being seen in Boston. He has very good hospital, very good neurologist, a lot of experts in leukodystrophies, but it doesn't get referred to leukodystrophy from the stroke side. The stroke docs were not thinking about this. This is a long time ago when stroke genes were not hot at the moment, 2004. So it will be different today, but still, it doesn't get to a diagnosis until 6 years later when it continues to be symptomatic, and this is then what happens in the following years. And again, the syndrome is wider than this. It's not just the white matter, it's not the ischemic lacunar strokes, it's not the large hemorrhages that can happen, or the vessel dissection. And for the patients that are listening to this and are here, would this happen to me? Most likely not, okay? So these are all separate syndromes that we don't see only in few variants. We see like all together coming together. So don't think about, oh, I have to be on the watch for all of this. We need to learn a lot about it, but that doesn't necessarily means that you have the entire. For example, this fusiform aneurysmatic dilatation or aneurysms do not happen necessarily in all patients. What is most common is these white patches in the white matter that I was showing you, the small vessel disease. You heard a lot about small vessel disease, neurovascular coupling today, and all these functions. I think that's one of our very good places to start if we're going to try to show that we have a mechanism of action for any drug that we try to get to these patients. So large and small vessel disease can give you bleeding, both in small arteries, microbleeds, or larger, and then you get a larger bleed with or without an aneurysm. And these patients are at risk of wall dissections, and you also heard from the conference last year, there was clinical guidelines that were like speared by the Italian work already, and then brought in with a lot of more experts across Europe and across the US. Cerebrovascular disease of the small vessel kind, you heard about this white matter injury, and then there's very different mechanisms underlying that white matter injury. This blood-brain barrier permeability, you just heard about modeling this, both in mouse models, all those blue, Persian blue stains are hemorrhage in the mouse. In the zebrafish, we saw the hemorrhages being watched live. And then we have iPS cells, blood-brain barrier modeling to do that. So that's a pretty good place to start. And then these small vessels are responsible for the lacunar ischemic strokes, which are a big burden in this patient population. And by the way, these are not, they're treated sometimes as lacunar ischemic strokes, and patients get put on anticoagulation, but we don't know if this hurts or helps. Particularly in PATMAL, they might even have a role, because the mechanism of these vessels clogging is completely different than what happens on a clot or atherosclerotic vessel. So increased risk of premature cognitive decline, we talk about these vessels are necessary for your neurons to fire, okay? No neuron can work if you don't have blood flow to the neurons, and I'll show you a couple of pictures on how that really can be looked at, and we heard and saw today in several models at the bench. So in terms of phenotype, I also think these kind of publications where groups of people, this is from the International Pediatric Stroke Organization, and there are many like this, I just took one example, are putting together that these genes need to be tested. If we don't get to diagnosis, we're not gonna know the full spectrum of the disease. And we have to make efforts for those that are not experts on genetic and rare diseases or a stroke to be able to recognize patterns. So we need to publish a lot about patterns of recognition and what kind of gene panels you can send. We're getting to the point that we're doing whole exome sequencing for almost everything, but there is still a role for genetic panels. And there are several, and they're different in every country, okay? If you talk about the US versus Mexico versus France, the panel of genes that you send for a patient are different depending on what your clinical labs have developed and validated for you. So these are just small efforts. There are many more papers, and I just put the ones that I have available a slide for. So identification of the genetic condition or the mechanism. These, thanks goodness, we're ahead of it. We do know what gene is involved here. We know you heard a lot about where the different mutations can be on the gene, and that may or may not sometimes correlate with the phenotype. Some of them do robustly. This is a very complex disease. I love this slide because it tells us that collagen is not just 4, there are many more. And it's not just one protein that is produced by one gene. It has to be these 3 proteins, 2 alpha-1s with an alpha-2, and this is COL4A1, just to make the triple helix. And then they start making these meshes, and they make the scaffold of collagen. And as you heard, we know now pretty robustly established that TGF-beta, which is one type of pathway that is, by the way, critical for all growth, development, and aging and cancer, is really involved in this. And this is where targeted, you remember when Doug was talking where do we need to treat? And Scott gave an example. If you have this inhibitor of the PI3K, if you do an MMP inhibitor, you will get an effect in your model. Can we afford that in a body? Can we afford that in a baby, in a child? For how long are you gonna treat them? We need to treat them for life. So I better be very targeted, so my side effect profile is very narrowed. That's one possibility. I better go very selectively to where the problem is, so nothing else gets bothered. That's ideal. So what Doug was talking about, like just fixing the little letter in the DNA that is causing the problem, if possible. And here is the genetics, and we have more. This is only COL4A1. You have COL4A2 variants. So getting to the characterization of the mechanisms and from cause to phenotype, this is what we call the correlation between genetics and the symptoms of the patients. And you have heard about the mouse models, and we do know some about where the mutations may be and how they may present, but in humans, by the way, we do not. There is a lot of fancy stuff that can be done in the human, and I want to put an emphasis on this because I think if we are doing a protocol like we did for leukodystrophies back in the day for another disease that got gene therapy approved, we have to start collecting data in a more consistent manner. This is a sequence that takes 2 minutes. It's called perfusion imaging. It's done at the same time that you had to wait for a contrast enhancement. Some people will say collagen 4 has blood-brain barrier leakage. I don't want gadolinium in the brain of my patients, okay? So I take it. That is not ideal. There is no ideal here. The idea that I'm trying to present is that we may be in front of what is going to be the only biomarker we can see an effect of a therapeutic in real time. And just to give you an idea, Cadacil is now starting clinical trials with T2 hyperintensities at the very research level, not the ones that we use here like in like T2 hyperintensities on the common scans. Cerebral amyloid angiopathy, there is an siRNA clinical trial started, Dr. Greenberg, that's putting a molecule inside the brain, trying to suppress beta amyloid, which is causing this. The first endpoint for efficacy and mechanism of action is functional MRI of neurovascular coupling for photic stimulation in the occipital cortex. You can read the paper that they did, 55 patients. The FDA let them start a trial in humans with this drug, and for readout, can you improve vessel function? They're gonna allow them to explore it with functional MRI. They don't have to wait 15 years to see if there is increased numbers of microbleeds. And that's what happens when you start looking at imaging. All the structural stuff takes forever to change. But we need more functional. We need blood-brain barrier permeability. We need microvascular function, and those are available, perfusion and functional MRI. And the best part is that you can do them in the mouse, you can do them in the zebrafish, you just heard. So now, if we do a drug, we put it in these animal models, we may be able to correlate the effect, your dose, to what you expect to see in this modality, just the modality in the patients. Would a drug get approved by this only? The answer is no. No agency will let you approve a drug if it doesn't improve the quality of life of the patient. I don't care what your brain fancy images look like. So you have to correlate these changes in the mouse, like a lot of people on the zebrafish were doing with behavioral testing that then correlates with the behavioral or the quality of life of the patients. Again, the patients are needed. We don't know yet quality of life surveys in COL4A1. Those are easy, more affordable to do, and you have a community that is widely engaged. You can go really fancy about this. We've done it in other mouse models. You heard about, at some point, the drug, when I manufacture the drug. So we have a clinical trial, yay, we're gonna do this. How do I know, how does anyone knows that the drug that I'm putting to one person versus another person is the same quality, has the same potency? They need to be tested in the dish before I give them to the patients. Every manufacturing batch needs to have something called potency assays. So everybody needs an assay to test the potency of the drug, and that's not a zebrafish, that's not a mouse. That should be the fastest, cheapest thing, and most robustly, consistently replicable assay you have in the disease. So we need this widely and now, so we can start doing, because when I write these pre-INDs for the FDA, there's a whole section of what are my potency assays. And it's not just the in vitro cultures in the human. I have to justify it with the mouse, with everything has to hint on my potency and how I'm gonna test the drug before I give it to the human. I think we're good. We do have cellular models that we can track. And the fastest will be a PCR and looking up TGF-beta pathway, if you ask me. But we have much more fancy, closer to the collagen disruption that can be done. So as we said, the characterization can go into biomarkers in humans. We can keep, and we should, keep looking at all the burden. We should also start looking at things that are a little bit more fancy. This sequence, by the way, takes 2 minutes, okay? It's not an add-on. Most hospitals have it for a stroke. So it's not that you also have to invent it in your scanners. This is available, but you do not necessarily scan all patients with this sequence. So you have to order it that way. And maybe we want to put this into a consideration for manuscript for what should it be a protocol, an imaging protocol for vascular leukodystrophies. Again, let's not do it only for COL4A1. There is many of them, and a lot of people will benefit from that. And then I told you prospective, retrospective studies are needed. This is also to biobank things. I'm going to put a shout on to remote visits. This is really highly embraced by the FDA. They're trying to find if there are wearables or remote markers like videotaping gait and doing analysis of gait, or things that are wearable and relevant to the disease that you can validate. And then finally, treatments. How do we do treatment? So before you go into any fanciness of drug development, I don't want anybody to forget that treatment is now with a standard of care, and we have a lot of tools to make people's and symptoms better, okay? And there are interventions such as you need to be taking rest and naps and breaks during the day. And that changes dramatically the quality of life of a person. And that's a letter from a doctor, okay? So I'm putting it in simple terms, but in reality, there is a lot of health that we can do and care now that improves the quality of life, and it might reduce the risk of stroke and cerebrovascular degeneration and white matter degeneration. And I like this because I use it for educational purposes, that 75% of your life and the life of an individual is usually outside the hospital, outside the rehabilitation acute. And we are doing very little here to do research about how this is happening and to bring therapeutics into that realm. So a shout to really think very proactively how to collect data from the home place. Are there gene therapies now being approved? There is more than 20 approved by the FDA in different disorders. This is what we call the bull eye, okay? And this is old already. It's like, I think, 2 or 3 years ago. And those are rare diseases. I took off so there is every single dot there is a clinical trial. And what is inside the gray zone is what is about to get to market, meaning it's going to be available for someone to use. So this is all rare diseases, okay? So you are not behind. You are on time, and we're just going, I think, faster than you could aim for. So there are different types of gene-targeted therapies. Very few words about this. You can target the DNA. You can add a gene, or you can edit a gene. We are in a disease that addition would not work. You know from Elizabeth that put in the map duplications are bad. So we know that adding the gene, not an option. taking down the gene, one allele, we heard today enough to say there will be a disease. It might be milder, but there might be a variant that is still pathogenic. Well, not variant, a disease phenotype that is pathogenic. Elegantly, we could try to edit the genome, and there are different ways to do that. But these are very challenging. These are state of the art. There is no in vivo vascular genome editing therapy in humans yet. Not a clinical trial. No one has been drugged with a CRISPR that goes to the vessels, okay? They've been drugged for CRISPR that goes to the liver. They've been drugged for CRISPR that goes to the eye and the ear. But nothing has been done in vascular. So we have to be humble that this may be several years, and I love the 5 to 10. This may be even further. What I think is much faster moving, and in fact, from saying identifying the variant to a clinical trial for a person of N01, Timothy Yu has done a lot of work. We have a regulatory frame for this, anti-sense oligonucleotides. As I was saying, this will take down the allele. That means, can you afford it? Is that pathogenic? Is that going to give you disease, or can you improve the disease? If I have a kid that is seizing 50 times per day, I'll try it, okay? Because as we said, there is no development if I'm seizing like multiple times an hour. So then it's all about the patient and how much risk they can tolerate and how much the benefit really means something to them. This is silencing RNAs. They're really fastly developing molecules, and they can be made to go to the vessels, by the way. And then microRNAs, which is also phenomenal work, but much more diffuse, and it's difficult to know which ones we need to do. And Doug is trying to take lead on some of the regulatory RNAs on collagen. And then small molecules. What is that? You just heard MMP9 inhibitors, PI3K inhibitors. Like, can we go after TGF-beta in one way or another? Absolutely necessary. Why? Gene therapy is not for all. Gene therapy, whatever it is, anyone you pick, will be for a subset or group of people with this particular presentation at a particular time with a particular disease progression. So we need other things that can ameliorate and slow down the disease. Maybe they're better than gene therapy, and they're small molecules. What I love about this arc is that when you get to here, you have to be very specific where you deliver your gene therapy because they're high risk, which means we have to develop the delivery method, how to get to the vessels, not the whole body. I'm not taking a pill by mouth and going everywhere. I have to take something by mouth, IV, or whatever that goes to the vessels. And those tools can be used to deliver small molecules. So that's what I like about this, working in multiple types of modalities. I heard that we have drug developers in the audience too, so in the manufacturing side. And then how do we get to the brain? Maybe this is very important. I think the eye is very accessible in this disease. And it's something that even if we have a therapeutic that is gonna make a difference, the ophthalmology field is going at like speed of light for gene therapy. So not to forget, whatever model you're doing, to please characterize your eye, because we will have the possibility to treat these patients in the eye probably before we can treat vascular. And then to get to the brain vessels, you can do it intrathecal, you can put it in the vein and try to get through all the vasculature. You can put it in an artery in the neck and get to the brain more directly. The ear, I don't think applies to this disease. And then the muscle, you can put intramuscular injections and stuff. I don't think necessarily that will work for us. But then there is a lot of considerations on how you do the selection of your delivery route. So this is another way to think about it. And I think I've been going through all of this, but from these like laboratory models and the essays that we were talking about to doing a screening of drugs, which is already ongoing for the disease, to clinical trials, and there might be gaps. We don't have this, we don't have that. So one of the things that I think will be absolutely worth an exercise for this community and a larger community is identifying the gaps. And we did this for every disease we have gene therapy today. So at some point, we sat down in a room and everybody participated, and we were able to identify priorities in the next 3 years, priority in the next year, to do the work and to collaborate to create that. The registries, for example, the biomarkers. I used 2 minutes to explain this. This is the first FDA-approved, EMA-approved gene therapy for a leukodystrophy, a white matter disease that is genetic. And they're published, so you can go see all the... This disease, again, is complex like this one, but not as complex as COL4A1. There is a lot more patients. And it can give you a spinal cord disease, adrenal insufficiency, low cortisol, or it can give you wiping white matter disease, which we call cerebral ALD. And this one is inflammatory. It's not like COL4A1. So a lot of not the same here, but the path to creating a therapy was, I think, is pretty similar for most of them. And as you can see, depending on what disease phenotype you have, either the brain or the spinal cord, you have now different modalities of gene therapy. So for the brain, we changed the bone marrow cells because they were inflammatory and we could treat that. And for the spine, the trial is ongoing and is putting AAV and intrathecal delivery of this gene. Much easier genetics and pathomechanism. Okay, adding the gene makes a humongous difference. That's not so easy for COL4A1. And we also knew that before we see anything on the MRI that you call a lesion, there was a change in the microvascular function, both blood-brain barrier permeability as well as microvascular capillary flow. So all these things we were hearing today are accessible with perfusion imaging. And again, this doesn't happen without a very engaged community and foundations that started the investigators to work together. And this is pretty much what we did. Took cells out of the patients, put the gene back. Sounds very simple. This is very complicated. Ouch, I have to give them myeloablation as a bone marrow transplant, okay? That's a very high-risk procedure, has a lot of toxicity associated with it, and then they get their own cells. Who was eligible for the trial initially? Kids that don't have a donor, okay? If I don't have a donor of bone marrow, I'm gonna die from the disease in 24 months. So it was an unmet need that was very high, and we could take this crazy risk of doing this. And by the way, the first patients treated were in France. They were not in the US where the trial got executed. What we learned was that it could work, and then a group in the US and a company took it on to do it. Kristen Duncan led the bone marrow transplant side. Florian Eichler led the entire study, and this is the cells that we think go from the bone marrow into the brain and correct the disease. And we still don't know, we're not very sure. We got approved by the FDA. This is what the lesions tend to look. So that's when they start. That's if you don't treat 2 years later. That's if you treat 2 years later. So we did really arrest the disease. We halted where we got it, pretty much. In 55 patients, there is always a but. This therapy comes with a side effect that we didn't know when we were doing it. We tried to do everything to not have this side effect, but it still happens, which is a pre-leukemia. So we're giving them, to a certain percentage of patients, a cancer. Still without involving the neurological function, so patients will choose this versus having the disease progress. Just I'm saying that because that's what's happening right now. The drug is commercialized. But there are buts. There is always buts. And I'm trying to say this because you are in a trial, and we enroll a lot of kids from around the world, and we didn't know what to expect, but we talked about all the things that could happen. And they do happen. And we had to transplant them again, and then you can cure this pre-leukemia. They don't get to a leukemia. But again, it's 2 transplants in a kid that is less than 18 years of age. So talking about the small molecules, we had other companies that were trying to use their technology and what they have. This is a group in Barcelona. The trial primarily enrolled in France, Barcelona, and Germany. As you can see, there is no FDA arm. There is now an FDA approved trial going on in adults. So this is a kids trial. And this is what happens when you have treatments. It ramps up. Like all these patients show up, you don't know from where, all over the world. But I just want to put the pictures of this particular trial, which I'm very close to it. And we have patients from all over the world. And this is at the same time the gene therapy trial is happening. And they're not necessarily coming from Europe. And that's what I'm trying to say. And that happens in rare diseases. The patients come from wherever they are. Your research should include patients from wherever they are. And that's what I want, consents that cross boundaries, registries that cross country boundaries, because we need them for therapies. Again, we know the blood-brain barrier is disrupted, and that's what this drug is supposedly doing, among other things. A PPR gamma agonist, and it has marvelous anti-inflammatory effects. This drug crosses the blood-brain barrier, which is good. You concentrate it more in the brain. And the trial just ended. And we are presenting in American Academy of Neurology the results, but this is a hint. Nexus is the name of the trial, and it decelerates the lesion progression, and it can arrest in some patients. So we met primary endpoint on this disease. It will be a drug that people can take at home by mouth in my home country where no gene therapy and bone marrow transplant happen in time, okay? Home country is Argentina, by the way. And they can take the pill and slow down the disease until they can access a more definite treatment if they haven't arrested by then. So I really like to have options. I really like to have molecules that are not so elegant as gene therapy because they're more realistic in the real world. There's plenty of monogenic vascular diseases. You're not alone, and they are all in the same conundrum that COL4A1 is. How do we get to the vessels? How do we make it targeted? It takes a large team You want to develop these things that we call delivery platform or vectors. That's a virus called adeno-associated virus. This is a lipid nanoparticle. You're probably familiar with these things. You need a lot of work on the models, so the mouse models, zebrafish models, cellular models. You need the clinical expertise. We were talking about this all along. And then you need to have a team of experts to do the clinical trial and the manufacturing. This is a little bit of what our gene therapy, vascular gene therapy group looks like. We have this mission, and it's one patient at a time with all in mind, because we don't know what patients we can treat now, but we are continuing to work in developing the therapies for everybody. You saw these pictures today a ton of times. The vessel coming in, these green guys are the smooth muscle cells, these guys are the pericytes, those are mural cells, endothelium. In this disease, we had to target probably both, depending on the stage, to correct the collagen. There is no vascular vector that is really robust. There may be a nanoparticle for endothelium here and there, but there is no AAV. So we developed an AAV that can do that. This is a human artery. That's a mouse brain. That's a mouse aorta. So we have translation to human specimens. These are arteries coming out of the operating room, fresh, put in a bioreactor, and we perfuse them with the different vectors, and it's superior to AAV9 and other vectors that we currently have in clinic. And we are using this for this disease. We are in front of the FDA on March 3rd with a pre-IND. This is a disease where kids don't know necessarily they have a mutation. It's ACTA2, and they start suffering strokes in a very rapid manner, and they can lose a lot of neurological function. The disease is caused by mutations in the ACTA2 gene, alter the cytoskeletal actin, a lot of similarities, and everything we've been hearing today about the models and the function. Those are the patients. I don't know how to go back, but I think it's this guy. These are our community, much rarer than you guys. We started with 14 patients, and now we have more than 90 diagnosed and around the world, and around 50 of them are participating in the clinical research, like retrospective and prospective study. The prospective study is around 14 patients at the moment. We do this always informing back what we are assessing in the clinical trial by the patient-reported outcome. And why? Because we cannot do testing that we can measure because we like to measure. We have to do things that mean something to patients. If not, the FDA will say, yeah, you changed the white matter hyperintensities in the MRI, but that doesn't correlate with anything that you show me matters to the patient. So we do this in parallel, and we redo it every couple of months. In this, we were doing a CRISPR base editing. We changed a letter, an A to a G, and we do it by delivering into this vector, this little enzyme called CRISPR base editor. Very complicated thing. We were doing high efficiency in the liver, not so high in the vasculature in the brain, significant in the heart, but this is sufficient, and this is the off-target. We got really lucky. We had to develop the assay for the off-target effects for gene therapy. And the off-target is a much larger work, and this reverses completely the vascular disease and the neurodegeneration, which is white matter disease as well. So we were able to do all of that. That's why we are in front of the FDA. Again, bringing perfusion. That's the same marker that we are going to propose to use as an exploratory. And the eye is going to be our window to the brain. And with this, there is many other diseases that we're trying to target in a multidisciplinary way. I will end with this, that is, yes, it's complex, we can do it, and it matters. It matters now because we change a standard of care, and it matters in the future because we will have therapies that can hopefully increase the quality of life. This is my lab and Mark Lindsay's lab, which is my partner in crime for all of this because he's the expert on smooth muscle. And thank you for sharing your journey, your knowledge, and your suffering with us is what keeps us going and what really makes things happen. And with that, I will end the day probably. It's enough today.
Thank you so much for this wonderful talk that is summarizing the whole day. And thank you also for the enthusiasm that you have given the speech, and also for being really clear about the risk and everything that means going through this journey.